Hello students, welcome to readmedpep Academy channel. Today in Chromosomal Basis of Inheritance part 1, we are going to discuss about the chromosomal theory of inheritance. G.J. Mendel in 1865 studied the inheritance of well-defined characters of pea plant. For several reasons, it was unrecognized till 1900. Three scientists, Hugo de Vries, Carl Correns and Erich von Schermang, independently rediscovered the Mendel's results on the inheritance of characters. Hugo de Vries from 1848 to 1935, Carl Correns from 1864 to 1933, and Erich von Schermang from 1871 to 1962. Various cytologists also observed cell division due to the advancements in microscopy. This led to the discovery of structures inside the nucleus. In eukaryotic cells, worm-shaped structures formed during cell division are called chromosomes. They were visualized by staining as colored bodies. An organism which possesses two complete basic sets of chromosomes are known as diploid. Here you can see in the eukaryotic chromosome where the nucleus is present in the cell which contains the chromatin material which condenses itself to form the chromosome. The chromosome is made up of the DNA. A chromosome consists of long, continuous coiled piece of DNA in which genes are arranged in linear order. Each gene has a definite position or locus on a chromosome. Here you can see the chromosome that is present in the nucleus, and the chromosome is made up of the deoxyribonucleic acid, otherwise called the DNA, and the portions of the DNA which code for a particular character are called the genes. These genes are hereditary units. Chromosomal theory of inheritance states that Mendelian factors, otherwise called genes, have specific locus or position on the chromosomes, and they carry information from one generation to the next generation. Now let us talk about the historical development of the chromosome theory. Chromosomes responsible for heredity have been found out by the cytological findings related to the chromosome theory of inheritance. Wilhelm Roux in 1883 postulated that chromosomes of a cell are responsible for transferring the heredity. Montgomery in 1901 was first to suggest occurrence of distinct pairs of chromosomes, and he also concluded that maternal chromosomes pair with the paternal chromosomes during meiosis. T. Boveri in 1902 supported the idea that the chromosomes contained genetic determinants, and he was largely responsible for developing the chromosomal theory of inheritance. W. S. Sutton in 1902, who was a young American student, independently recognized a parallelism or similarity between the behavior of the chromosomes and the Mendelian factors during gamete formation. Sutton and Boveri in 1903 independently proposed the chromosome theory of inheritance. Sutton united the knowledge of chromosomal segregation with the Mendelian principles and called it the chromosomal theory of inheritance. What are the salient features of the chromosomal theory of inheritance? Somatic cells of organisms are derived from the zygote by repeated cell divisions called mitosis. These consist of two identical sets of chromosomes. One set is received from the female parent, otherwise called the maternal chromosomes, and the other from the male parent called the paternal chromosomes. These two chromosomes constitute the homologous pair. Here you can see what happens in mitosis. The first stage is the interphase, and in the prophase stage there is condensation of the chromatin material resulting in the formation of the chromosomes. In the pro-metaphase, the chromosomes are well formed and try to come towards the equatorial plane. In the metaphase, in mitosis, the chromosomes get arranged in a linear fashion in the equatorial plane. In the anaphase, the microtubules are formed and the microtubules try to separate the chromosomes on either side. And in the telophase, nuclear division occurs and cytokinesis occurs, resulting in the formation of two cells. Chromosomes retain their structural uniqueness and individuality throughout the life cycle of an organism. Each chromosome carries specific determinants or Mendelian factors, which are now termed as genes. The behavior of chromosomes during the gamete formation, which occurs during meiosis, provides the evidence to the fact that genes or factors are located on the chromosomes. Here you can see the meiosis. Meiosis consists of two parts, meiosis 1 and meiosis 2. Meiosis 1 is reduction division. In the interphase, the chromatin material tries to get condensed in the prophase and forms the chromosomes. In the metaphase, there is accumulation of the chromosomes in the equatorial plane and the formation of the microtubules. In the anaphase 1, there is separation of the chromosomes to either side or the either poles. And then in telophase, there is formation of the nucleus and cytokinesis occurs, resulting in the formation of two cells in meiosis 1. In meiosis 2, after the interkinesis stage, there will be condensation of the chromosomes. In metaphase 2, there is accumulation of chromosomes in the equator. In anaphase 2, there is separation of the chromosomes by the microtubules. And in telophase 2, there is formation of the nucleus and the cytokinesis occurs, resulting in the formation of 4 cells. So, meiosis results in the formation of 4 cells. Meiosis 1 is a reduction division and meiosis 2 is the simple mitotic division. Now, let us compare between the genes and the chromosomal behavior. Around the 20th century, cytologists established that generally the total number of chromosomes is constant in all cells of a species. A diploid eukaryotic cell has 2 haploid sets of chromosomes, one set from each parent. All somatic cells of an organism carry the same genetic complement. The behavior of chromosomes during meiosis not only explains the Mendel's principles, but leads to new and different approaches to study about heredity. Consequently, exchanged at a holiday junction, moving the branch up or down the DNA sequence. This is called branch migration. Now, let us compare the chromosome and the gene behavior. The chromosomes that are present on the left side are the homologous chromosomes. They have the same genes and same order. You can see the allele A and C as the nitrogenous base. On the right side, you can see chromosome 2 and chromosome 3. The chromosome 2, the left side, you can see the homologous chromosomes of the chromosome 2, and the right side, you can see the homologous chromosomes of chromosome 3. If you take the chromosomes of the chromosome 2 and chromosome 3, this is called the non-homologous chromosomes. And the genes that are present are different. For example, you can see the allele small a and the nitrogenous base is T here instead of C, that is present in allele capital A. So, what are the differences between the Mendelian factors and the chromosome behavior? Alleles of a factor occur in pair, whereas chromosomes occur in pairs. Number 2, similar or dissimilar alleles of a factor separate during gamete formation, whereas in the chromosomal behavior, the homologous chromosomes separate during meiosis. In Mendelian factors assort independently. In the chromosomal behavior, the paired chromosomes can separate independently during meiosis, but the linked genes in the same chromosome normally do not assort independently. Here you can see the number of chromosomes in different organisms. Left side is the organism and the right side is the number of chromosomes which are diploid. Adder's tongue fern Ophioglossum has 1262 chromosomes. Horsetail Equisetum has 216 chromosomes. Giant Sequoia 22 chromosomes. Arabidopsis 10 chromosomes. Sugarcane 80 chromosomes. Apple 34 chromosomes. Rice 24 chromosomes. Potato 48 chromosomes, maize 20 chromosomes, onion 16 chromosomes, Haplopappus gracilis 4 chromosomes. Thomas Hunt Morgan in 1933 received the Nobel Prize in Physiology or Medicine for his discoveries concerning the role played by chromosomes in heredity. Now, what are fossil genes? Some of the junk DNA is made up of pseudogenes, the sequences present in that once called working genes. They lost their ability to make proteins. They tell the story of evolution through the fossilized parts. Here, these fossilized genes give the evidence of evolution. So, fossil record composed of physical remains of species. Geological distribution of organisms indicating the common ancestral species. Homologous structures implying similar genes. Comparative embryology implying similar genes. So today in chromosomal basis of inheritance part 1 we discuss about the chromosomal theory of inheritance and the different historical implications. Thank you. Kindly subscribe, like, share and comment the channel readmedpep Academy. Log on to www.readmedpepacademy.com for MCQs. Our Facebook ID is readmedpep Academy. Our email is readmedpepacademy@gmail.com. Thank you very much.